The topic of this presentation is cell structure and function. This is William Raja, Assistant Professor from Bishop Weber College, Trichy. Before going to the title, let's learn the outline regarding the cell structure and function. Fluid mosaic model has the lipid bilayer and integrated proteins also. In general, there are some cellular organelles like mitochondria, lysosome, ribosome, endoplasmic reticulum, Golgi apparatus, vacuoles, chloroplast. And the main important organelle is, as we all know, that is nucleus. Nucleus contains of the genetic material, and in chromosome contains of 80% cystone protein and 20% non-cystone proteins, and nucleolus is the formation of ribosomal RNA. And this is the outline we are going to see now. The first one, cell theory. What is cell theory and who introduced the cell theory, and what is the cell dogma? All these things we will learn. And in dogma in cell biology, we say the dogma of cell biology is transcription and translation. Gene is converted into mRNA, that is the process of transcription, and mRNA is converted into protein, that is the translation. That is the central dogma of cell biology. And cytoskeleton is responsible for rigidity to the cell and consists of microfilament like alpha and beta tubulins. The properties common to all cells and cell size and shape, prokaryotes and eukaryotes, what are the difference between these two cells? At last we'll end with cell junctions, what it is all about. And before moving to learn about the cell theory, the history is more important. The two scientists, Anton von Leeuwenhoek and Robert Hooke, they contributed much in cell biology. Anton von Leeuwenhoek improved microscope. Robert Hooke especially Koch cells. He observed Koch cells. He observed many cells including Koch cells. So these two scientists contributed much in this field in the 16th century. In the year 1850, Rudolf Virchow, he proposed that all cells come from existing cells. He was the one and proposed at first. And cell theory, there are three important points regarding cell theory. All organisms consist of one or more cells. Cell is the smallest unit of life. All cells come from pre-existing cells. So we can observe under microscope, light microscope, and other microscope we will discuss later. And light microscope can observe living cells in true color, and the magnification up to approximately 1000x resolution, it can be visible. And the microns could be 0.2 to 0.5. Electron microscope, there are two important electron microscope. We generally say TEM and SEM. That is nothing but transmission electron microscope and scanning electron microscope. The TEM, transmission electron microscope, we can get 2D images using this TEM, and using SEM we may get 3D images, and these are the images we usually get it from SEM and TEM. And cell structure, all cells have an outermost plasma membrane, genetic material DNA, and cytoplasm with the ribosomes. So all cells have an outermost plasma membrane, as we discussed. In a nutshell, we will see what is the structure and what is the function of it. So it has a phospholipid bilayer with the embedded proteins. It controls, this is the function of outermost cell membrane. It controls what gets in and out, or what goes out, and gets the signal also. Genetic material like DNA. In eukaryotes, DNA is within the membrane, that we call it as nucleus. In prokaryotes, there is no membrane around the DNA. That's why it is called as nucleoid. All cells have cytoplasm with ribosomes. Cytoplasm has fluid area inside outer plasma membrane and outside DNA regions. So in between, outside of the DNA region, cytoplasm will be present. Ribosome is the site of a protein synthesis. Cells need sufficient surface area to allow adequate transport of nutrients in and wastes out. So round cells will always be small. So in general questions, we may ask why cells are so small. The round cells will always be small. And let's learn about prokaryotic and eukaryotic cell structures. Prokaryotic cells have smaller and simpler in structure compared than the eukaryotic cells. So prokaryotic cells do not have a nuclear bound organelles. So the structures, prokaryotic structures, contain of all these organelles. There are certain marked capsule, flagella and pili. These are the marked organelles which may be present in some prokaryote and may not be in all prokaryotic cells, but these are the general organelles which is present in prokaryotic cells. First one, plasma membrane, cell wall, cytoplasm with the ribosomes, all we discussed earlier, and nucleoid, capsule, flagella and pili. So this is the structure of a prokaryotic cell, and flagella will be there for a moment. And this is the observed under TEM, transmission electron microscope. In the case of eukaryotic cells, the cells have nucleus, ribosomes, endomembrane system, mitochondria, cytoskeleton. Mitochondria is the powerhouse of the cell. I hope all you know very well. And the cytoskeleton, as I said earlier, it is responsible for rigidity to the cell. It consists of microfilament like alpha beta tubulins. So this is a structure of eukaryotic cells. Nucleus is the most important organelle in which genetic material will be there. The functions could be: DNA directs the activities of the cell. So the structure, nuclear envelope will be there, 2 phospholipid bilayers with a protein lined pores will be present, and nucleoplasm means the fluid of the nucleus. Mitochondria, as I said earlier, this is the powerhouse of the cell. It is helpful in synthesizing ATP, and there are 3 important pathways involved in ATP production. The first one, glycolysis pathway, Krebs cycle, electron transport systems. So the structure could be approximately 1 to 5 microns. The outer membrane, inner membrane, intermembrane space, matrix, all these organelles, main organelles will be available. and this is the structure of mitochondria, and it is observed under TEM, transmission electron microscope. Vesicles have small membrane bound sacs. So examples: Golgi and endoplasmic reticulum transport vesicles, peroxisome, lysosome. So in peroxisome, where fatty acids are metabolized and where hydrogen peroxide is detoxified, and that is the role of peroxisome. Vacuoles have membrane sacs. It is generally larger than vesicles. Vacuoles generally larger than vesicles. So examples could be food vacuoles and contractile vacuoles. Cytoskeleton, the function is it gives cells internal organization, shape and ability to move. So it is helpful in shaping and the movement also. That's why I have said earlier, it's responsible for rigidity to the cell, so it will have the proper shape because of the cytoskeleton. The structure, it is interconnected system of microtubules, microfilaments and intermediate filaments. All are proteins. So this is the structure of a cytoskeleton. In microfilaments, the most important protein is actin. The thinnest cytoskeleton elements seems to be rod-like structure. Microfilaments composed of globular proteins, actin. I like actin. Microfilaments will show enable cells to change shape and move. So intermediate filaments, was the role of intermediate filaments in cytoskeleton is it is present only in animal cells of certain tissues, and it is the fibrous proteins joined to form rope-like structures. So which could provide the internal proper structure. Cytoskeleton, microtubules, the microtubules which is present in cytoskeleton, that have long halo tubes made up of tubulin proteins. Cilia and flagella, which is used for movement, cell motility, it will be useful. Microtubules wrapped in an extension of a plasma membrane. Plant cell structures, and these are the important organelles. Certain structures may be found in plants but not in animal cells. So like chloroplast, central vacuole, other plastids, vacuoles and cell wall. The function of a chloroplast is a site of photosynthesis. So the chloroplast, which is involved in the process of photosynthesis, because of this organelle, the plant produces its own food, but it's not in the case of animal cells or animal. There are two outer membranes, thylakoid membrane system, chlorophyll in granum. Granum is nothing but stacked membrane sacs. Stroma, it is the fluid part of a chloroplast. So the diagram is depicted here. It is visible, and grana and stroma is highlighted. Plastids or vacuoles in plants. Chromoplasts contain colored pigments like carotenoids. So in plants there are other pigments also available like xanthophyll, chlorophyll A, B, and including these carotenoids. Amyloplasts is helpful in storing the starch. So the function of vacuole is the storage area for water, sugars, ions, amino acids and wastes. It may contain poisons to protect against predators. So it is also helpful to protect, for the protection purpose it is helpful. The structure of vacuole could be large membrane bound sac. It occupies the majority of the volume of the plant cell. Cells interact with their environments and each other via their surfaces. Many cells are protected by more than the plasma membrane. So the cell wall, which is the most important organelle also, it provides structure and protection. If the plant doesn't have cell wall, there won't be a structure. So it provides structure and protection. It never found in animal cells. It is present in plant, bacterial, fungus, and etc. Structure could be, it wraps around the plasma membrane. It is made up of cellulose and other polysaccharides. It connects by a plasmodesmata. So it seems to be channels. So in the cell wall there will be a channel. So you can see in the diagram. So that is a channel in the cell wall. That is the purpose of a plasmodesmata. It will make a channel. Cell junctions, it's also most important organelle. Plasma membrane proteins connect neighboring cells. That's also called as cell junctions. In plant cells, plasmodesmata provide channels between cells. So there are three important types of cell junctions in animal cells. The first one is tight junctions, occurring junctions, gap junctions. Tight junctions have membrane proteins seal neighboring cells so that water soluble substances cannot cross between them. Concurring junctions have cytoskeleton fibers join cells in tissues that need to stretch. For stretching purpose it can be useful. So it will be present between heart, skin and muscle cells. Gap junctions have membrane proteins on neighboring cells linked to form channels. So this is all about the different junctions. So with this basic information, I like to finish my presentation. Thank you for your patient listening. Thank you all.